I wanted to briefly introduce myself, but first just welcome you all to the webinar. And I'm Nancy Stack, President and Founder of the Cystinosis Research Foundation. We're thankful to our panelists, Stephanie Cherqui, Jordan Janz, and Barb, Kulyk, for being on the call today. You'll meet them shortly, and they're the people you really want to hear from. So we'll have plenty of time for that a little later on the call. Just a little bit for you. On this call, you'll have the ability to send questions with the Q&A feature. If we have time, we will try to answer or ask and answer your question, but we did receive so many questions. We'll see how many we can answer in the time allotted. You'll be muted during the conference, but we will unmute everyone at the very end of the session to say goodbye. The chat feature is also available for you to use. So thank you for joining this call and for your support and your involvement with Cystinosis Research Foundation and your involvement in our community. I wanted to share a little bit about CRF since I just don't know a lot of you out there. So just a little bit of background about CRF. My husband and Jeff and I have two adult children, Alex and Natalie. And Natalie is 29 years old and she has cystinosis. We started CRF in 2003 with the sole purpose of raising money to fund research. When we started the foundation, there were only a handful of researchers interested in cystinosis. Research studies were scarce, and because of a small population of patients, large grants were literally unheard of. We knew that the only hope for a brighter future for our cystinosis community was to fund research. For us, research is hope. Hope that a cure will be found for cystinosis. We started as one family, but we were quickly joined by other families from all over the world. Together, we're determined to change the course of cystinosis and to find better treatments and a cure for this disease. To date, CRF has funded 200 multi-year research studies in 12 different countries. We're currently funding 38 active studies in areas focused on the kidneys, eyes, the neurological complications of cystinosis, and more recently, studies focused on myopathy, muscle wasting, and bone issues. CRF research has led to the discovery of a slow-release form of cysteamine, which was FDA approved in 2013. It's now known as Persispi. CRF research has led to this FDA-approved clinical trial to test Stephanie Cherqi's stem cell and gene therapy work. The research we fund helps our entire community of children and adults. And we like to say that no matter where you live in the world, and at any moment in the day, you can always be assured that there is a CRF researcher working on unraveling the mysteries of this disease. CRF is the largest fund provider of cystinosis research in the world. We issue grants twice a year. In fact, we just funded $2.6 million in new grants in July, and we will announce new grant awards in December. The research never stops. In addition to the research we fund, we have an annual conference known as the Day of Hope, which is the largest annual gathering of families, patients, and researchers in the world. We had to cancel the conference this year because of the pandemic, but we are definitely planning on having the conference in 2021. And in order to increase our odds of having the conference and being able to gather during this time, we've scheduled it for later in the year. The conference is now scheduled for August 26th to 28th in Newport Beach, California. It will be our first summertime conference. We certainly hope you can join us, and of course, we'll be sending out more details as time goes on. Together, we created a global research community that's improving the lives of adults and children with cystinosis, and we thank you for being part of our quest. Now, today's stats on this call, I thought you'd be interested in knowing that as of this morning, we have 273 participants, and those participants include adults with cystinosis, grandparents, parents of children and adults with cystinosis, sibling and other friends and families, clinicians, researchers, and people from industry. We have 14 countries represented, and this is quite impressive. So I'd like to name the countries because we welcome all of you. We have people from Australia, Belgium, Brazil, Canada, France, Germany, Ireland, Italy, Mexico, New Zealand, Sweden, Switzerland, the UK, and the US. Thank you all for being here. Now, the most important part of the conference is hearing from the panelists. And these are three very important people in our community and in our lives. So first, I'd like to introduce Jordan. He is surely one of the bravest people We know he was the first patient to volunteer for the trial to risk his life for all of us. He's a pioneer and an amazing person. Jordan will share his perspective as a patient. And Jordan, why don't you just say a few words about yourself right now?
Hi, I'm Jordan Janz, and I was diagnosed with cystinosis at 8 months old. As most of you know, I was the first person to go through the stem cell trial. And I'm just over a year now after the stem cell transplant.
Well, good. Thanks, Jordan. We'll be asking you questions, so stand by for those.
Okay.
Next, I'd like to introduce Barb. She is Jordan's mom, and she was with Jordan during his time at UCSD during several, I guess it was 3 or 4 months that you were there, maybe in total or even longer. But she's been was his caregiver during the trial and has a unique perspective as a parent and caregiver. Barb, would you like to say a couple words about yourself?
Hi, thank you for inviting me today. I'm Jordan's mom, as you all know. He's my third of 5 children. I have 3 grandchildren, and I live in a small little town in Canada, and I'm just happy to be here and that we had this opportunity, so thank you.
Thank you, Barb. And now we have Stephanie Cherqui, and what can we say about Stephanie? She is a brilliant and dedicated researcher. She was part of the team in Paris that discovered the cystinosis gene. She moved to the United States, and we were fortunate to meet her and persuade her to work for us and with us. We knew she had a passion for cystinosis and was determined to find a cure for cystinosis. We started funding her in 2007, and to date have funded almost $6 million for her stem cell work. She has dedicated her career to cystinosis research and to our adults and children with cystinosis, and for that we are eternally grateful. Stephanie, please introduce yourself, and then you can go ahead and share your presentation.
Thank you very much, Nancy. Thank you to you and to the Cystinosis Research Foundation for organizing this event, because it's so important to stay connected in this challenging time. I can believe this is a very special event for me, because presenting with Jordan, Barb, and Nancy, it's very special. I mean, I cannot believe that it's been a year now that the transplantation occurred. And it has, and Nancy and I, with Barb and Jeff, we attended the transplant. It was a very special moment that we will never forget. So I will present today the initial results that we have for the phase 1/2 clinical trial. So I hope you see my slide, right, Nancy?
Yes, I can
Okay,
see
good.
it. I assume everyone can.
All right, so I have to start by my disclosure. Right. Okay, so as you know, cystinosis is due to a gene called CTNS that is expressed in every single cell of the body. It encodes a transmembrane protein, cystinosine, that it allows the export of cysteine out of the lysosome. And the storage of cysteine in patients with cystinosis lead to this very characteristic cysteine crystals in all the tissues and lead to a multisystemic disease. I will not go into detail what is cystinosis. I mean, you all know what is cystinosis, and today we will really focus on the clinical trial. So I'm trying to be as short as possible to have some time to answer a question after. So the therapy that exists today is the supportive therapy. So people take medication to prevent some complication, but also the cysteamine therapy that really act on the cysteine storage. The approach we want to take is to go at the origin of a disease, which is going to the gene. And we want to do what we call gene therapy. So correct the gene or bring a new gene into the cells. The issue here is that, as I said, the gene is expressed in every single tissue. So to bring a new gene to the tissue or cells, you need what we call a viral vector, like HIV vector or herpes virus vector. I mean, this vector, we make them safe, and we put the healthy gene, and they can bring a gene to tissue or cells. But here, because the gene has to be replaced in every single tissue, we can just not only give a viral vector like that. We need to have a vehicle to bring the gene in every single tissues. And this is why we are taking the bone marrow stem cells, because the bone marrow stem cells they are pluripotent. So this means that they are able to turn into some tissue cells or to blood cells, and they can target the tissues. And so against all odds are the hematopoietic stem cells that are reside in our bone marrow were the one that had the best effect on the cystinosis in the mouse model. And I will not go into the preclinical studies because, again, we don't have that much time, and I really want to focus on the clinical trial. But I mean, along the years, we have seen that using the hematopoietic stem cell, we were able to, which give rise to blood cells, we were able to decrease the cystine content in tissues and to prevent the development of a disease. So how do we do that? So the approach in the patient is to take the hematopoietic stem cells from the blood after mobilization of the stem cells into the blood. Then we take the stem cells and we gene correct them in a culture plate. So that's why we call that ex vivo gene therapy. So we use the stem cells and really we modify them ex vivo, so out of the body. And to do that, as I said, we use a lentivirus vector, which is a self version of HIV. And this lentivirus vector contains the CTNS gene, a healthy copy of the CTNS gene. So we add the stem cells with a virus vector, and at the end of the day, you end up with a mix of some cells that are gene corrected and some cells that are not gene corrected. And after, to be able to, for the new stem cells to integrate and repopulate the bone marrow and replace the disease cells in the bone marrow, we need to do a chemotherapy. So like that, we remove the host stem cells that don't have the corrected genes. And after, when we infuse them, one-time infusion, the stem cells know that they have to repopulate the bone marrow, and so where they will go, and they will stay in the bone marrow for the rest of the life of the patient. And the good thing about that is that after, this is create a reservoir of healthy cells for the rest of the life of the patient, because these cells are able to integrate into all the organs and provide a healthy copy of a protein to the different tissue. And again, most of you have seen along the years how this works, and that, for a matter of time, and will not go to the details, but we have characterized the mechanism of action, etc. So again, to just make sure that this is clear, I just did an easy schema. So this is what is happening in the culture plate. So you have the hematopoietic stem cells that are missing the gene and the protein, and you have a lentivirus vector that infects the cells, and the DNA, RNA, but the genetic material of the virus go to the nucleus of the cells and integrate into the genome of the cell. So like that, the cells are gene corrected for the rest of the life of the cells. And because they are true stem cells, they will stay and multiply into the bone marrow. So this is the principle. So for the clinical trial, we have it really takes a village to reach that point of a clinical trial, but it also takes a village to conduct a clinical trial. So we are three main principal investigators. So myself, Dr. Barb, that most of you know, he has been in the field of cystinosis for many years, and Dr. Ball, who is the director of the bone marrow transplant at UCSD. But we have many other physicians, and you can see the name, and you will recognize several experts on cystinosis that Dr. Troner, Dr. Mack, Dr. Dohill, and we have also Dr. Grimm in the consortium. All these doctors work together to be able to design the best clinical trial and after to analyze the data. So this study will be to test the safety and efficacy of the treatment, and a total for the phase 1-2 clinical trial, which is what we are doing now, there will be 6 patients maximum. The primary goal is to test the safety, but also we want to test the efficacy. but by checking, the cystine content into the white blood cells or tissues, and to also determine the clinical outcome of the disease. So we will have, as I said, 6 patients. It's a staggered design. So there are 3 cohorts of 2 patients. The cohort 1 and 2 will be adults, and the cohort 3 will be adolescents if the risk assessment looks good. So, so far we have completed the cohort 1, and we are about to start the cohort 2. But it's really 1 patient at a time, and I think we will discuss that during the questions, because in the world of gene therapy, the FDA wants us to be very careful, so we have to enroll 1 patient at a time. So it's very important. So the patient have to have a good organ function, for especially good respiratory function, and this is because of the chemotherapy. We want to make sure that the patient can go through this step, but can be challenging. So once a patient is selected, there is an informed consent and screening. So they have to pass screening where we test the tissue function. And then the patient has to go through a lot of evaluation, which is what we call baseline evaluation, to determine and evaluate all the tissue function before transplantation, because all this will be retested after transplantation to make sure that the treatment is beneficial for the patient. So we do the kidney function, eye function, muscle, neurological function, quality of life, respiratory, endocrine testing, and we do also rectal biopsy to be able to take some tissues from the patient, so we can determine the cystine content and the engraftment of the cells into the tissues. And we also do skin biopsy and in vivo confocal microscope to be able to see the crystals in the skin and in the eye. So it's very important to realize that all this is demanding, and but it's because it's a phase 1/2, so we don't know where to look. We don't know what we are, on what we will have a beneficial impact if we do. So we have to test a lot of parameters. So the patient come into San Diego when this clinical trial start, and after the baseline, and so we do a mobilization of the stem cells by 4 days of G-CSF-64, and after, the patient is hooked up to a machine for like 4 or 5 hours to get the white blood cells from the patient. One bag of apheresis will stay at UCSD as backup in case something wrong happen. And 1 bag will be transported to UCLA, where they will the gene correction of the stem cells will occur. And then the product is cryopreserved, shipped back to UCSD, and then the patient goes through busulfan conditioning, so the chemotherapy, which is 4 days only, but it's a full myeloablation. So we try to remove as much of stem cell as possible, and then we have a one-time infusion. So we had, so far we transplanted 2 patients. Both of them were males. One is 20 years old, as you know, and one is 46 years old. They were both heterozygote for the 57 kb deletion. So the first patient had no kidney transplant, but he was in stage 3 chronic kidney disease at the time of the transplant. The second patient had 2 kidney transplants. So I know that some question ask, can we do the stem cell therapy if you have kidney transplant? Yes, you can, and we have already have 1 patient with actually 2 kidney transplants. Both of them were taking cysteamine and cysteamine eye drop before the transplant. But what you have to realize is that 2 weeks before the transplant, the patient stopped cysteamine and cysteamine eye drop, and we want if everything looks good, we keep them off cysteamine for as long as possible, and for John, and it's been a year that he's not on cysteamine. So for the product, so the stem cells, when they gene correct, to determine the efficacy of gene correction, we measure what we call the vector copy number. here, and it's the number of vector per cell. And for the patient 1, we were at 2, and for the patient 2, we were at 1.27. So anything above 1 is really good. So, I mean, we were lucky that the 2 first cell products were nicely gene corrected. So, to show you. All right, so there was, for the 2 patients, no serious adverse event or adverse event related to the product, so to the cell, to the gene-corrected cells. There were actually no serious adverse event. And most of the adverse events that were reported was due to either the disease or the conditioning, so the chemotherapy. And actually the most side effect that the patient was reporting the most detrimental was the mucositis. So what happened is that when you start with chemotherapy, the stem cells really start to drop slowly, and at 10 days, they are really going to 0. Then they start to go back up, and this is what we've seen for both patients as expected. All right, so now let's check. So now the cells have been transplanted, and now we are checking the patients, what's going on in the patients. And as I said, one parameter to test how, if there is a gene correct cells that are gene corrected into the patient, we take the blood of the patient and we measure the vector copy number into the cells and the CTNS expression. So the expression of the healthy CTNS protein. So here you see the vector copy number. So at the beginning of a transplant, usually it really represents the product. So for the first patient, we have something around 2, which was expected. But you have to realize that when we transplant the CD34 stem cells, you have a mix of progenitors and true stem cells. The one that really stay into the bone marrow and repopulate the bone marrow are the true stem cells. So, and the progenitors have actually died around 6 months. So at that time, you expect a decrease of vector copy number. So this is what we've seen as expected. But after that, you want this to stay stable, and which is actually what we see. And actually, we even see a slight increase with time of a vector copy number. So this is good. And here you see the CTNS expression, again, a decrease, but now going back up. All right, so after what we did is measuring the cysteine content in granulocyte and leukocyte. So here you have a baseline measurement of the cysteine content in the first patient, and this dropped after in the first 3 months. And after the patient, as you know, live in Canada, so he had to go back in Canada, and the sample were shipped to UCSD. And so we don't know if it's because we were manipulated differently or there is something different with granulocyte and leukocyte, because we saw an increase on granulocyte cysteine, but the granulocyte actually are included in the leukocyte. But what is surprising, the leukocyte, they stay at around 1, which is actually the targeted therapeutic, therapeutic target for cysteamine, and what the parents' heterozygote value. So we are not too sure why there is a discrepancy between granulocyte and leukocyte, but again, anyway, what we want to know is really what's going on in the tissue, which is really what's the most important. But we have to use and find the best biomarkers. For patient 2. Again, at 1 month, we have, I remind you, the cell product was around 1.3, so we have a vector copy number in the blood at 1.4. The granulocyte cysteine, in this case, the leukocyte cysteine were higher than the granulocyte and didn't show much decrease, but the leukocyte that was much higher showed more than 60% decrease, and the value are even better now. So again, there is a discrepancy between granulocyte and leukocyte. Leukocyte seems to be a better biomarker, but again, what really matters is what we have in the tissue. Again, that's what I really want to make the point of. So to do so, we have developed some exploratory endpoint, and one of them is using a confocal microscope to visualize this skin crystals. And we developed in my lab using a lot of patients and a lot of you maybe on the call today, you participated to a study, so we could develop this tool and quantify the cystine crystals in the skin. And for patient 1, we have seen, so the baseline number was around 2,200, and at 3 months post-transplant, it was at 1,500. So because the patient couldn't come back because of a pandemic for so long, and just right now, just came back and actually left the day before. So we're just starting to collect the data. So unfortunately, I don't have the latest data because we are just starting to analyze them. But at least I have some hint on this one, and the trend of cystine cystine crystals in the skin continue to go down and seems to be even lower than 1,000 now. So for the urine volume in the patient 1, so the baseline was at 4 liters, and this was decreased at 3 and 7 months, and at 9 months was a little bit higher, went back to higher. For the kidney function, so this is measured by the glomerular filtration rate and serum creatinine, and as I said, the patient was in a stage 3 kidney dysfunction before the transplantation, and what has been lost in the kidney are not expected to come back, but what we want is something that stays stable. And so far, we had a stable GFR and serum creatinine, and if we compare to the slope, before the transplant, so in the last 4 years, there was a tendency of decreasing, whereas now we kind of reach a stable line. So we will see with a new data, and we have much more specific markers for kidney function that we are currently analyzing to make sure that the kidney function is stable. There is also if you knew Jordan before the transplant, you can now see the difference because it's very striking, and it actually teaches us something that we didn't think. It's like all the patients have blonde hair and white skin, very pale, and it's because actually the cystinosine and it's because the melanin is very low into the patient. And there was one publication in 2012 that showed that cystinosine itself, so the protein itself, is involved into the synthesis of melanin. So it seems like after the transplantation, it's strongly because you can see that the skin become darker and the hair color become darker. It seems like the protein is present and make the job of producing the melanin. But now this is what we have to test objectively, and we added new testing in the protocol because of what we observed. So now the patient 1 stays off cysteamine for a year now. He was taking 52 medication. Now he's taking 25. And have a less episode of vomiting, so which is really good. But I mean, I'm not insisting on that because Jordan, as you know, is here and will be able to speak about his experience. So the patient 3 has been enrolled and completed baseline, and the product has been characterized and manufactured. So it will be ready to go very soon. I just want to finish by acknowledging all of you, all the cystinosis committee, all the parents, all the family, all the volunteers. I mean, the volunteers to this trial are heroes. It's a first in human, so there are many risks and they don't know if there is any benefits. So I really thank all the volunteers and all of you for being here and supporting Jordan, me, Nancy, the foundation Barb, I mean, we did an amazing journey since we started this project in 2007. And I cannot believe it's already so long. But I think it really takes a village to go that far. So, and a lot of support and funding. So thank you to all of you, to the foundation, to all the sponsors, like CERM and NIH, in addition to obviously the Cystinosis Research Foundation, to all our collaborators, the staff, the medical staff, all the doctors, and our partner AvroBio, who will really take this project to the next phase and to all of the patients. All right, thank you so much.
Thank you, Stephanie. That was great, and we have questions being typed in as you spoke, but I'm going to start with some of the questions, and I'll start with you, Stephanie, since it'll be a follow-up to what you just presented. And here's, we had a lot of these questions about gene therapy and when it will be available outside the United States. Do you have any insight or information about that?
So I don't have a specific date, but AvroBio is very, working very hard to try to bring that to the next phase, as I said, and the phase 3 actually will be in several sites, including in Europe. So they have already started to work on that. So we work in parallel to be able to start the phase 3. So maybe before I'm even done with phase 1-2. So as soon as we start, it will be available in other countries. But I don't have a specific date. It's impossible to give a specific date, and depends on so many parameters.
Well, you mentioned the phase 2 and phase 3. So when do you think you'll be done
with
that?
We're right now in phase 1-2. And the phase 3, this is when it will be open in other countries. But it's not, there is no date, but we hope this will be as soon as possible because, as I said, the phase 3 might even start before I'm done with phase 1-2. Okay.
And when do you think you'll be done with phase 2?
I don't know. No, I mean,
next year, do you think?
It really depends on the data. I mean, you have to, especially with COVID, we are not at the level we wanted to be at this stage because we couldn't get, the patient back. We had also a gap. So in the enrollment of patient. So we need to determine, the real consequence of a transplant. And if the data are really good, more the data will look good, faster we will go. It needs to be safe and we need to have enough time. The FDA wants to see at least, 2 years of follow-up before starting a phase 3, I guess, for safety purposes, so.
Okay, and then a question you touched on about the 6 people participating in the trial. Can those people come from outside the United States?
So in theory, yes. And I mean, you see that, patient 1 is actually from Canada. It's just that, it's a lot of commitment because you have to stay here, in San Diego. You have to come every 6 months for the baseline study and follow up. But at the time of transplant, you have to stay about 2 to 3 months in San Diego. So when you are out of the US, it's just, more complicated. This said, also the priority so far, will be to the US patient, because we are in the US. But again, if everything goes fine, this will be open to oversight very quickly. I mean, again, maybe another year or so.
Okay. And another one about the trial. Can you please explain the selection and decision-making process for participants during this part of the trial and subsequent phases? I think you touched on that, but is there anything you'd like to add?
So first of all, for the cohort 1, we didn't enroll patients who were homozygote for the 57 deletion, and I will not speak about that now. But we did some study to make sure that missing the gene next to CTNS was not impacted the effect of the stem cells. So we couldn't enroll before I was done with the study the patient with homozygote for the 57 KB deletion. Now we can. So first we selected patients who were not homozygote for 57 kb deletion. But even the other, they have to have good organ function, and this is not determined by the study, it's determined by the chemotherapy, so by the bone marrow unit, most of the criteria. They want to make sure they can go through the chemotherapy, and that's determine who can or cannot participate. But in the same time, we have a list of participants, we take them in order, and we try to take them in order. But again, we select one by one. So, we have identified patient 4, but we don't know who is patient 5 or who's patient 6. We cannot do that yet, so it will be one by one.
Okay. And in order to get the final FDA approval, which is what we all want, how many more patients should receive the treatment before? Will it be the 6?
So FDA approval for what? For the whole,
as a therapeutic treatment.
Yeah. So that's so it will be after phase 3. So it will be it cannot be before. So it's not. It will be much more patient than the 6 patients.
Okay.
And the obviously the safety has to be good. The data should show efficacy, but it has to be after phase 3.
Okay. And back a bit to the genetic mutations. Someone wrote, I would like to know what type of genetic mutations the mice had that you used in your experiments, and if you have any indication that the gene therapy might have different effects depending on the type of genetic mutation.
It is a very, very good question. I wish I knew. So we don't expect any difference, but as I said, the only one we were not sure was the 57 kb deletion, because it removed the SHPK gene that can be involved in macrophage differentiation. So if you know the mechanism of action, it could have been a concern, but we have done the study showing that it shouldn't be a concern. So, but except that, there is no genetic mutation that we don't know anything about if there any genetic mutation would beneficiate more than others, or if there is any impact. This because the mice, as you mentioned, has all the same genetic mutation. It's actually a complete deletion of part of a protein, so it's completely non-functional. But it's always the same mutation. So it's the first time we're really going to the real world with many different mutations, many different people, many different genetic backgrounds.
Now we'd like to skip over to Jordan and ask him a question. Jordan, I think you have to unmute yourself there. Yep. Okay. So the question we had the most was this big picture question for you. How are you doing? And how are you feeling?
I'm doing really good. I notice like little changes, but they're gradual. And so it's hard for me to notice them, but over time, I definitely notice changes. Like I have, I feel good. My energy is definitely coming back, and I feel like my energy wasn't as good as before, I guess. And I think that's because of the chemo I went through. But it's definitely getting back. I'm definitely getting better with energy wise. I don't have the smell from the pills anymore, which I really like, because I don't have people saying, what's that smell when I'm by them. And actually, when I got home, I went to work, and a couple of the guys I work with turned around and smelled me and said that I smell normal. And for me, that was like, wow, that's awesome. So that was pretty nice. But overall, I'm doing really well.
That's so good. So you're back at work, and do you feel like you can work full time, or?
Yeah, I got home, it was January 3rd or 6th, I think. And then I went back to work at the end of January, so full time.
And let's see, we have a question that came in. How does Jordan feel differently after the gene therapy compared to before? That's from Colleen. Can you compare?
Yeah.
Before and after?
So before the gene therapy, I guess, I was definitely more tired, but I'm always the person that needs my sleep. But if I get enough sleep, I'm good to go for the day. Whereas before, if I didn't get enough sleep, I was tired throughout the day. Yeah.
So you have noticed some difference.
I've noticed some difference. It's more, my mom notices more difference. Me, it's kind of gradual, and it's hard for me to notice. But.
And Barb, maybe you'd like to comment on what Jordan just said, and he mentioned you as maybe observing a difference.
He's a little quicker to get going in the morning. Jordan's made pretty much our entire family late our whole life, so he is not late anymore. He is up and he's going. I think it's because he's not nauseous in the morning as much. He still has days where he is, but the pills and the amount of pills and having to eat, and now he can grab a granola bar and a juice box and run out the door like the rest of the kids. Where before it was always making sure we had a large meal and time, and it was letting him get ready for things at his own pace. Where now he can do things more on the fly, which is kind of neat to see.
And have you observed the lack of smell too, or the odor that our kids have from histamine? Is that impactful?
As a mom, like all of us on here, we don't find that odor foul. It's my child, it's your child. I go to these conferences and smell the kids, and I think they smell beautiful. It's just part of who they are. I didn't notice Jordan until I think it went away. Then I could smell, he smelled like my other kids did. You know how your kids have that smell in their hair that always smell kind of like sulfur? I guess that's gone. So I notice the things as they leave, maybe not so much as we lived with them. I think that more of a psychological change for Jordan, maybe rather than physical. Like I see that he's confident and he's happy and he's got color. I mean, I'm always feeling his face to see if he has a fever, and he just has color. It's just, he looks good.
Amazing.
Yeah, I'd like to add on to that too. I noticed like when I got home, I felt way more confident about myself, because before when I would hang out with people, I would always be wondering, are they smelling my pills, or do I look sick because I'm so pale? And now I just don't think about that.
Well, what do you think about your darker hair, Jordan?
I definitely like it. I kind of miss my blonde hair, but I definitely like this hair. When I got home, when my hair started to grow back, I walked upstairs the one day and I have a mirror at the end of my stairs, and I looked in the mirror and kind of was like stunned by myself. I thought that I looked a lot like my brother and thought that was weird.
Oh my gosh. That's a big change. A big change. I guess we won't know if everyone has that change, but it certainly has been a big change for you. Well, good. I probably will go back to Stephanie, because we have a lot of questions again for you, Stephanie. So let me cover some of them. What are the potential risks in a stem cell transplant?
So when you, it's mostly because of the chemotherapy first. I mean, it's when you have no immune cells, you can have infection. So that's why you stay in the hospital for 2 weeks after transplant. So they monitor your temperature all the time. Monitor anything that could happen to make sure there's no infection. You can have also consequences of chemotherapy, because I know that there is a question about infertility. It can lead to infertility. At this dose, it's not 100% sure, but it has a good chance. So the patient can freeze their eggs, or sperm, and use them after. But that's an issue of a chemotherapy that can occur. Gene therapy, we never know. It has its own risk, because we add a new gene, so there is always this risk. But I have to say that in this kind of ex vivo gene therapy, there are hundreds of patients who have been transplanted, for many disorders. And so far, I knock on wood, there is no death that could be, or very serious adverse event due to the gene therapy itself. But it always can be a risk.
So a lot of people have asked about fertility, and we're getting questions now about it again. So the chemotherapy will cause infertility. Most likely.
Most probably, yeah.
And so then if someone's participating in the trial, they can go ahead and harvest for the women.
Yes.
How does that work?
So I mean, I'm not, a fertility doctor. So some patients, before when they're enrolled, they can, before the transplantation, they can freeze, their eggs or sperm before going through a transplantation. And I have to say that, thanks to Cystinosis Research Foundation, we're helping some of the patients to cover that during this phase 1/2 clinical trial.
Okay, that's good. And thank you. Will every patient have mouth sores from the chemotherapy?
Yeah, so one of the main adverse events that occur is, and that are painful, is the mucositis, so the mouth sores. But actually, for patient 2, it was been less because we knew beforehand. So it was treated preventively. So it was in a much less degree than the first patient. It's because now, we have a clinical coordinator, in our in the trial, who works really with me, and we try to advise the patient as much as we can. And so I think for second patient, it was much less than the first one because we knew before.
Thank you for that. And we have a question here that, and I'll read it the way it came in. I feel that the researchers or the program is focused on those that do not have many negative complications from the disease, such as muscle weakness, myopathy. When or are they going to tackle this problem or this reality, actually? These adults are in desperate need of this type of transplant. Don't want it to be too late.
Yeah, and I completely agree on that. So one of the main issue is the muscle weakness, and why is because of the respiratory function. And one of the patients didn't pass the screening because the respiratory function was not good enough at the time of the screening. So they maybe the patient is now working with a respiratory doctor to try to improve this, and this is due to the chemotherapy again. So as I said, most of the exclusion criteria is not due to the gene therapy or the stem cells, it's due to the chemotherapy that is needed, and there is no way around it. So the bone marrow unit said, no, it's not meeting the criteria for chemotherapy, so we cannot do it. But we know, and actually we were very, or the physician on the trial, we really wanted to do this patient because we want, to see, and maybe if there is muscle weakness, we might help. We don't know that. There is no way to know, and I think we were very disappointed when the patient didn't pass the test. Now there are many groups who works towards going around the chemotherapy for a good engraftment. So this is something that's coming in the future. It's not here yet, not good enough yet, but that's the future. So hopefully the chemotherapy will not be necessary anymore. And then there will be, most of the patient can be enrolled at that time. But at the time here, we still need it. And that's what makes the criteria more strict.
Well, do you think actually that after this 6 patients and when it opens up to more patients in different centers at some point with AgroBio taking the lead there, that won't be a problem, that, because people who get chemotherapy are often very, very sick, but it still is helpful?
I wish, I could say yes. I don't know, I don't know where. I think AgroBio has also one of the program is to try to find a way around the chemotherapy. So we are actively working on it. Will that be ready for phase 3? I'm not sure, I cannot speak for them, but I would be surprised that it would be so early, but maybe.
Maybe. Okay. And then this is actually for both Jordan and for Stephanie. Do you think the treatment will help the light sensitivity, or will it just keep getting worse? So will it stabilize it? Will it improve it? And Jordan, you can actually speak to what you're experiencing as well. Have you noticed a difference with your photosensitivity or any change in your eyes and your vision?
Definitely noticed a difference. My eyes aren't as sensitive to light. Like when I would wake up in the morning before and I would look at my phone, I'd have to turn the brightness down because I wouldn't be able to look at it. And now it's fine to look at the phone in the morning. And when I came back for testing 2 weeks ago, I was in San Diego. I had eye tests and I noticed my eyes weren't as sensitive to touch. Like before, when you would touch my eyes, they would instantly start watering and be irritated. But now when I did the eye test last week, I still didn't like it, but I noticed it was way better than before.
Okay. And Stephanie, do you have
So I mean, as I said, so I didn't see the data. I didn't even have a chance to talk to the ophthalmologist. So we just collect the data. So I cannot talk too objectively. But after he said that, I tried to sneak into the ophthalmologist notes, and it seems like there are some objective tests and that measure the sensitivity and even eye watering. I think he was talking, and it seems like it's decreased. So it seems like there is objective measure, but I don't want to talk until I really speak with an ophthalmologist and be sure. But yeah.
Good. Well, that's great to hear both of your perspectives. So Jordan, we're getting a lot of questions about water drinking. Do you crave the water like you used to crave it, or did you prior to the stem cell transplant? What are you doing with water now?
Prior to the stem cell transplant, yes, I was always drinking water. I had a big jug of water I would take to work and try and drink one of those. And I was probably craving water all day. And now I'm not craving water, but I still try and drink lots of water to try and keep my kidney healthy. And just overall, I'm not craving it anymore.
Stephanie, have you noticed anything in your data?
I don't know about the craving of water, but I didn't see any other data on that we are going to analyze. So we have a very sensitive and cutting edge, testing for the kidney function, and I don't have any other data yet.
And then we have a lot of questions since it's been 1 year for Jordan. Jordan, everyone wants to know which medicines you're taking, and if you are off cysteamine for Cysview or Cystagon, and also are you taking any eye drops?
So I was on Cystagon until I was about 12 years old, and then I was in trials in Chicago and was on Pro-Cysview from 12 to the stem cell transplant. And I haven't been on Pro-Cysview for a year now, but I am taking my kidney pills still. So I'm taking potassium, sodium, carnitine, and then I'm just taking like a couple of vitamins, and that's basically it now.
So since we usually quantify the pills, what was your high level of pills prior to the transplant? Where are you now in terms of numbers of pills?
I think it was like 55, and now it's in like around 25, but a few of those pills are vitamins, so I don't like necessarily need them, but they are good for your health, so I do take them. And then eye drops too. I haven't been on eye drops for a year either.
Good. Okay. Let me see here. We're getting a lot of questions typed in before. Maybe Barb, why don't you, I'd like to ask you what the hardest part of the trial was for you when Jordan went through all of this. Maybe you could explain how you moved here and lived here for a number of months.
I think the hardest part was trying to be the caregiver for Jordan, but still try to be mom to the other 4 that were at home. My husband was there with the kids at home, my older kids. You can say I was Jordan's caregiver, but there was a lot of people all over the world being my caregiver. At the end of the day, when I would be strong for Jordan all day, and then I would, need to talk to somebody myself, because it was hard. And I think the hardest part about being a caregiver to someone you love that has chronic illness is you can't fix them and you can't take the pain away. And it's just basically sitting there in the same room. So I think throughout the times when Jordan had his mouth sores and we were in the hospital, I would think he didn't want me there, but I'd leave and I'd get a text of where did you go? And I'd come back and, it was basically just sitting there and being present. And sometimes there's nothing you could do any more than that, than just be there, and there's just no right answer. It's kind of what works for everybody. But to come home and see the hope and the quality of life, and, I would say it was worth it for us.
Thank you. I'm going to try to read right now, so bear with me for a second. We are almost up against our 1 hour, but if people want to stay on, the panelists agreed that we could go for another 5 or 10 minutes. So you can stay on. The webinar will not just close down. Let me see. Oh, well, we do have a practical question here. Maybe Stephanie can answer. How much do you think this therapy will cost once it's outside of this clinical trial?
So there is no answer that I can provide at this time, but this costs a lot of money. Right now, for example, the Process B costs a lot of money, but it doesn't cost a lot of money to really generate, to make the pills. Here, the treatment itself costs a lot of money. But let's say, I don't know, I just give a number, but let's say it's $1 million, to pay for that. I think the health insurance would prefer to pay a one-time $1 million if we see that it's really efficient and prevent the use of cysteamine than to, keep paying the Process B for many years. So I think the cost wouldn't be an issue for cystinosis patients, again, it's still a lot of money to produce, but I think it would be for the insurance, the health insurance, they would prefer this therapy.
Okay, some unknowns still. We have a question and about the age of patients and the next cohort, because we started with 18 or older, then it was 14. So can you speak to the age of people participating, and when will infants and toddlers be able to participate? And actually, what is the best age?
And so, that again, it will be very complicated to answer. I would say, if you do it early, the way I would say it is like, any time after 5 would be great to avoid any tissue defect. I think the prevention is better than the repair, because, I don't think we can really repair things that is lost, but, but I think we can keep healthy what is still there. So if you do it as early as possible, it would be nice. But to go to the children, you have to first show a very good safety and efficacy in adult and adolescents. So if everything goes fine, we'll start adolescent in the phase 1-2. So this means that for phase 3, they will definitely enroll adolescents and maybe children. We don't know yet.
Okay, thank you. Let's see here. We've covered a lot of these. We have a question from someone that says, our son's doctor doesn't seem interested in this and says it's too experimental. Do you have any advice for finding doctors in Australia that may take and have more interest in this?
I think that if a patient is not interested, I think, it's their choice, and I think, we shouldn't push them otherwise. I think that the good thing is, AVROBIO have already some clinical trial going on in Canada and in Australia. So when it will go to AVROBIO and phase 3, they will know what doctor, and they will have doctor in Australia. So if it will be, they will have some advice at that time.
Okay, and let's see, we've got Jordan, has your appetite changed?
Yes, my appetite has definitely changed. Before the stem cell transplant, I was constantly eating all the time. And I think it was because I had so many pills to take. And if I didn't take them on food, I would throw up. Also, right now I'm not really hungry all the time, but I think it's because I feel like it's because my kidney is storing more. It's not just going right through me, whereas before it would just go right through me and I would always be hungry. I'm not as hungry as before, but I still have a big appetite to some extent.
And are you working out too? I think you are. And did you do
Yeah,
that before?
Actually, I was started again. I haven't in quite a while, just because of what I went through. I just kind of took a little bit off, but I started again a couple weeks ago in San Diego, and now I'm 2-week quarantine, so I have been working out every day at my house.
Good. And Stephanie, a question for you that came in. Can a patient have a kidney transplant after having the stem cell treatment, if necessary?
I mean, yeah, I mean, I would, yeah, I mean, I don't think that would be an issue.
Okay. And then to Jordan again, from Aaliyah Walker. She wants to know, Jordan, do you feel stronger and faster than you did before the trial?
It's a hard question. I'm gonna say no. Like, I feel probably about the same. It just depends. If I start working out like I did before, then I'm gonna feel stronger and faster. But if I don't work out like I did before, then I'm not gonna feel stronger and faster. So I think it depends what I do for activity.
Thank you, and those are all good questions. So I guess we'll kind of wrap it up now. We're about 5 minutes after the hour. Stephanie, and this was a question that came in, at what point in time do you think you can call this a cure, or will it be stopping the progression of cystinosis, or could it be both?
As I said, for people who know me, I cannot use this word cure, and I don't think I will be able to use it ever. I am very cautious, again, it's a first in human. We need to see with a long term, and I think with also a lot of patients, because patient can react differently. As you saw in the presentation, also the quantity of gene correction can depend, from one patient to another. But I think we need to look, and if if the efficacy is really proven on the long term, I mean, we could call that at least a better treatment. That's the most I will be able to do.
So we wait.
Yeah.
And we've only
we
had
wait and see. And
2 patients so far. And by the way, is patient number 2 also off of cysteamine?
Yes.
And patient number 3 will be transplanted probably sometime in November, early November?
Yes, I mean, soon.
That's the plan.
Yes.
Yes, okay. Okay, I think, let me just see. I did have one more question for Jordan, maybe. Did you notice any increase, and you've addressed this a little bit, in your muscle strength since the procedure, and did you have problems at all with swallowing difficulties prior to the treatment? So if you did, are there improvements?
I've always kind of had problems with swallowing, and I don't know if it was because of cystinosis or because I just had a bad gag reflex. Like, I still have a bad gag reflex, but swallowing, I would say, is probably about the same from as before, and muscle strength, I would say, is about the same. And then I'm hoping to get back into my daily workouts and gain a little bit of muscle strength, but it's about the same as of right now.
Okay, good. But any of the panelists like to add anything to conclude the session at all?
No, I think I think it was really good to, as I said, to be connected and get V7 going. I think I see that there are a lot of questions, so it's kind of not enough time to go through all of them. But I think Nancy, you agree. We can do that again another time, and we will try to through the Cystinosis Research Foundation to keep everybody updated on the advancement of this trial.
Yes, and as you mentioned, Jordan was just there. You collected a lot of data, so at some point in time, you'll have that information too, and we'll keep everybody posted and updated on the next patient and what happens there, and then any information you can share with us, we would of course love. So in conclusion, really, I thought we'd be able to unmute the group, but we can't do it, so I'm sorry about that. We don't have that ability at this time, but we're just grateful for all of you who have participated in the webinar. We hope you found it interesting and full of information. We hope that we'll see a lot of you next year at the Day of Hope, because Stephanie will present at that time as well. And then Jordan will be almost 2 years post-transplant. So all of you, please stay safe and healthy during this challenging time for the entire world. And thank you, and we will be in touch. Thanks for all your questions and for participating today.